Hello, my biologist. Welcome to another biology with Miss Judd. I want you to take a moment and reflect on our bodies. Now, we are not the same size that we were when we were 7 years old, and we are not the same size that we were when we were 3 years old. In fact, it takes a lot for a baby to grow into the human that we are today. Today, we are going to explore this process that drives cellular and developmental biology. So, we're going to be talking about mitosis, understanding cell division, and then look at the cell cycle and how is it regulated? How can disruptions lead to cancer? And then we're going to go through fertilization and cell differentiation and development, looking at how a single cell can become a whole complex organism. It sounds like we need to talk to Miss Judd in the corner. Hello, everybody, and welcome to our last lesson in Unit 3, Cell Growth and Division. In this, we are going to be talking about how a cell grows and divides. One of the main problems of cell size is a matter of surface area to volume ratio. So if we look at this problem, we can think of cells like a cube, and it gets larger and larger and larger. As a cell grows, it needs more materials to cross its membrane. Traffic is comparable to this movement of materials such as nutrients, water, oxygen, and waste across a cell membrane. As a cell grows larger, its volume increases. The number of traffic lanes or the amount of surface area to cross does not keep up. Materials cannot enter or leave the cell as quickly as necessary. So if a cell grew too large, it would not have enough relative surface area to get the amount of oxygen or nutrients into the cell and waste products out. So what we want to do is maximize our surface area to volume ratio. In order to do this, we divide our cell. So we produce two daughter cells. When a cell divides, there are a couple things that need to happen. We need to make sure we have 2 times the amount of organelles, 2 times the amount of DNA. So we have to replicate the DNA before cell division. This avoids the problem of information overload. Cell division solves the problem of increasing the cell size by decreasing the cell volume. So it makes more cells instead of larger cells, more cells instead of the cell continuing to grow. And that keeps the surface area to volume ratio high. Cells go through a period of growth after they divide to make sure that they have the amount of materials they need. Reproduction is one of the most important characteristics of living things. For a single-celled organism like this bacteria, simple cell division is enough for reproduction. It can allow populations to grow very quickly. This kind of cell division is a form of asexual reproduction, which is the product of a genetically identical offspring from a single parent. Bacteria can reproduce asexually, so they reproduce quickly in the right environment, kind of like at school. In both, they're asexual, which means they have the exact same genetic material as their parent. The opposite of asexual reproduction is sexual reproduction. Humans and other animals grow and repair their bodies through cell division. But to reproduce, we have to use sexual reproduction. Sexual reproduction involves the fusion of 2 separate parent cells coming together, which means we have 2 separate DNAs coming together. While asexual reproduction involves separation of a cell, sexual reproduction involves fusion or joining of cells. The offspring here are not genetically identical. They are different from the parent because we have 2 different pieces of information coming together. Characteristics that make an organism well adapted to its environment are critical for survival. When resources such as food and space are unlimited, it can be helpful to be able to create a very large number of offspring in a very short amount of time. When environmental conditions are stable, it may be useful to have offspring that are genetically identical to the parent. When finding a suitable mate might be difficult or impossible, it could be helpful to only need one parent to reproduce. When the environmental conditions are changing and new combinations of traits, they actually may promote survival. So it's useful to have offspring that are not identical to the parent. In sexual reproduction, we create relatively fewer offspring because growth takes more time. You need to find a mate, and in changing environments, this genetic diversity can be beneficial. Offspring may be less well adapted to current conditions, but if the conditions change, it could become well adapted. Most organisms use only one strategy. However, there are some organisms that can use both, depending on the environment. So, yeast can undergo cell division to reproduce asexually, but under certain conditions can enter a sexual phase. Talk about this process of cell division. How does one cell become two cells? All cells store genetic information in chromosomes, and most prokaryotes have this singular circular chromosome found in its cytoplasm. In eukaryotic cells, DNA is packaged into multiple chromosomes. There's more DNA because it's a more complex cell. In this multiple chromosomes, we start with our DNA double helix. That's where our genetic information is. That double helix wraps around histone proteins and nucleosomes. This coils, so imagine just continuing to twist it. And then finally, we have our sister chromatids with a duplicated chromosome held together with a centromere. This X-shaped replicated chromosome is what we're going to be working with in our cell cycle. So there's a series of events that a cell goes through in order to create two new cells. It is broken into three stages: interphase, which is G1, S, and G2, and then mitosis, or the M phase, and then cytokinesis, the cell splitting. In interphase, this is the period of growth before the cell actually divides. Cells actually spend most of their life in interphase. Cells do most of their growing in G1. It begins when mitosis is done and DNA replication begins. So in S phase, DNA is synthesized or replicated. Chromosomes are replicated. In G2 phase, many of the molecules and cell structures, like our mitochondria, are produced. We duplicate those organelles. This is usually the shortest phase of the cell cycle. We're essentially getting ready for our M phase, which is mitosis. This phase is much shorter than interphase, and the result of this phase are those two daughter cells. We go through mitosis, which is where the cell's nucleus divides, and then mitosis leads to cytokinesis. The cytoplasm divides, and two cells are formed. This M phase, mitosis, can be broken into 4 steps. First, we have prophase. In prophase, the nucleus condenses and chromosomes become visible, so that DNA coils up to create those chromosomes. The spindles form, and our nucleus starts to break out. The nuclear envelope breaking is the very end of prophase. What this means is that the DNA is condensed down and it's safe now, so we can open up the nuclear envelope. We then move into metaphase. In metaphase, the centromeres of each duplicated chromosome line up the center of the cell. Spindle fibers connect to the two sides of the chromosome. Each chromosome carries identical genetic information. We have our chromatid, our centrioles, and our centromere. This is important because this spindle connects to my centromere, which means that once it's connected, I can pull those sister chromatids apart. These sister chromatids, now called chromosomes, move along the spindle fibers to opposite ends of the cell, and that's anaphase. Finally, once the DNA has moved to opposite ends of the cells, we start to reform that nuclear envelope. So the spindle breaks down, the cell membrane begins to pinch together. Now, at this point, I have one cell and two new nuclei. To end this process, this M phase, I go through cytokinesis. That's my final step of cell division. This usually takes place right at the end of telophase. In animal cells, the cell membrane pinches together to form two daughter cells that can pull apart. In plant cells, a cell plate forms in the center of the cell. This plate usually develops into membranes and a wall forms. Now, what I have at the end here are two cells that are genetically identical. Let's talk about how we control this cell cycle and regulate the cell cycle. Scientists have found that many proteins regulate the cell cycle. It's controlled by these proteins both inside and outside the cell. The internal regulators respond to events inside the cell and let the cell cycle move forward only when certain steps have already been completed. For example, some regulatory proteins function to keep mitosis from starting until the chromosomes have been duplicated. Another regulatory protein keeps the cell from moving into anaphase until the spindle fibers have attached. External regulating proteins direct the cell cycle to speed up or slow down. So, growth factors are an important group of external regulator proteins. Growth factors stimulate the growth and division of cells. Other external regulators have the opposite effect, causing cells to stop or slow down their cell cycles. Stopping or slowing a cycle can be just as critical as stimulating growth. Stopping the cell cycle can prevent excessive cell growth and keep body tissue from disrupting another. While many new cells are forming almost constantly in multicellular organisms, cells are also dying. Some die just due to normal wear and tear. Other cells are programmed to die. This process is called apoptosis. Apoptosis plays an important role in structuring the body during growth and development. So, if old skin cells did not die and shed, it would become too thick and lose some of its properties. Apoptosis is totally normal and actually beneficial to many organisms. In fact, apoptosis is necessary for multicellular organisms. It allows organisms to fully control which cells continue to grow and divide and which do not. If a cell is old or damaged, apoptosis allows the cell to be destroyed rather than continuing to grow, divide, and use resources that other healthy cells need. When the cell cycle is not controlled, that's actually cancer. And I'm sure we've all heard this term or have an idea of what this is. Cancer is actually a disease of the cell cycle. So we have this diagram here, and it's showing cancer. This is a tumor. So when these cells grow without control, it builds up, and that's called a tumor. One cell started dividing too fast, and there is no control over the cell cycle. This rapidly growing mass of cells can be harmful to healthy cells. Cells need resources to grow and divide. When cancer cells grow uncontrollably, they not only displace healthy cells and tissues by taking up space, but they also use up resources that normal cells do. To take a closer look at this, we have first a cell beginning to divide abnormally. Then cells produce a tumor and start to displace normal cells and tissues. And then third, cancer cells move to other parts of the body. That closes us out for Unit 3, Lesson 4. You guys are awesome, and I'm going to send it back to regular Miss Judd. Thank you so much, Miss Judd in the corner. That was so helpful. We are going to be working with this in class. We really need to make sure that we have our notes done. Make sure you bring those to class so that we can work with this and see how this applies to our real life. You guys are awesome, and as always, make good choices.